So thank you so much for the kind introduction, Keen. It's really good to be here, and thanks to the organizers. I'm happy to kick off this seminar series. So I'm going to tell you today about some of the adventures that me and my lab have had in trying to understand sort of gene regulation and understand the central dogma using single molecule sequencing methods. So what do I mean when I'm saying single molecule sequencing? Well, what I mean by this is usually either PacBio sequencing as shown on the right or Oxford nanopore sequencing as shown on the left. Now, for many years, these were circus curiosities at best, where you could maybe do it for one study, but it would cost way too much money or be it was just logistically impractical. But now, you can actually do this for human genomics and to understand what's actually happening in human disease or on tissue samples that we actually get. So it becomes really applicable. On the left, I'm indicating that you can get 100 gigabases of sequencing data at about 98% modal accuracy from nanopore for $1,000. And you can run as many as 24 or 48 flow cells simultaneously on the instrument pictured there. That's a picture from my lab. And then on the right is the PacBio sequencer, which is probably the most accurate sequencing on the market today, even more accurate than, say, Illumina sequencing, like at least 3 or 4 nines of accuracy from the sequencing reads. And you can get about 100 gig from them as well for about $1,000 and fewer flow cells at a time. I do like to disclose that I do have some patents which have been licensed to Oxford Nanopore. So how did we get here? Well, initially, what we did was we were using a hidden Markov models to try and figure out nanopore sequencing. Because what's happening is you have a DNA or RNA molecule that's sitting in a pore. And as that molecule is in the pore, you're feeling the molecule with electrical current. And by measuring that current, you can get bases, but you're not measuring one base at a time. You're measuring multiple bases at a time. In this case, I'm showing you an example where we're measuring 3 bases at a time. This is from an earlier paper of mine from 2012. But the beauty is that as the DNA is advancing 1 base at a time, you know what the previous 2 bases are. So if I started with GTC, I could now be TCA, TCC, TCG, or TCT. That means I can reduce the problem. Instead of having to look for the entire space of all triplets, I can now reduce the problem. This is a commonly solved problem using something called Viterbi decoding that has been used in electrical engineering for years and was developed in part to try and solve problems of noisy cell phone signals. And by using this, we actually were able to do base calling from nanopore sequencing. And this is from about 10 years ago. This is from the so-called R6 chemistry of nanopore. And what you're seeing, the image where each of the horizontal lines is a sequencing read, and the places where you see color are places where there's a mismatch between this and the reference. The places where you see dashes are places where there's a missing set of bases, and places where you see purple carets are places where there's insertion. There's bases that shouldn't be there. You can see this is really noisy. It worked. It worked. It was amazingly, it worked. But it was really noisy. This was at about 70% accuracy. Since then, we've come a long way. Base calling has shifted to something called recurrent neural networks, where you're basically putting in training data and teaching, I'm sure you guys have all heard of AI and OpenAI, and we all use ChatGPT, and it's fun to talk to, and it makes us feel good and gives us lots of compliments. But what this is doing instead is this is taking training data where we give it DNA sequence that we know what it is, and then we say, here's the electrical signal we observed. Learn from this. Learn from this and figure out what the sequence must be when I then give you some other new sequence. And you can train with a neural net over and over. You can train it on these DNA sequences to figure out what the sequence looks like. With this, with improvements here, and with improvements in the chemistry, which I don't want to undercut, but I don't have time to discuss at length here, and where they're changing the different pore, they're changing the speed of translocation, they're changing the mode of protein. Nowadays, and this is as of last year, this is the accuracy we see from nanopore. So this is actually at a larger scale than the previous one. This is about 1,000 bases, whereas the other one was 100 bases. And you can see that these reads look very gray. They're very straight across. We have very few errors in individual reads. There's a couple reads that clearly had some problems, but overall, we're getting pretty accurate reads. And this is where I say that on a modal, the mode of accuracy is about 98% accurate per read. We weren't satisfied with this in my lab. My postdoc, Courtney Hall, wanted to see, well, okay, the beauty of this instrument is that it's about the size of a USB stick or like a Snickers bar or a small stapler or something like that. And you can plug it into a laptop. So maybe I can use this for distributed sequencing. But I might want it to be really accurate. I don't want to send stuff out for Sanger sequencing anymore. I don't want to figure out what the DNA sequence is. I just want to be able to take my amplicon and read it out on my laptop. but she said, I want it to be really accurate. I want it to be like 5 nines accuracy. And so she decided to work a little bit more on this method where she's incorporating what's called unique molecular indexes. So what she does is she does a first round of PCR where she puts on sort of a random DNA sequence onto the ends. Then she amplifies from there where each molecule has the same, after you do that, the subsequent amplification, each molecule had that same initial molecular index. You can see here from the colors, I have the sort of and blue, I have the blue and orange, or I have the sort of here. And each of those pairs of, I have multiple copies of the same molecule. So now when I sequence it, I can collapse those down. I can say, okay, I see these same molecular indexes multiple times. Those should all be the same molecule. So if they disagree, they're lying. The original molecule was something like this. I can use that for correction. We can also do stuff where instead of just sequencing the molecule once, we can actually sequence the second molecule. DNA is double-stranded, so I can sequence the first strand, and then I can capture the second strand, sequence that too. And Oxford Nanopore has some chemistry that does that they call duplex sequencing. So when Courtney did this, she could get near perfect accuracy. So I told you that before the modal accuracy was about 98%. For this stretch here, she found that the simplex reads, so sequencing one strand with no polishing, she could get about 0.957% error, or it's like 99.05 accurate. When she used her polishing tricks, she got to only an error rate of something like, I guess, 0.003. Or 99.997% accurate. This is incredibly accurate. This allows us to actually be able to do sequencing for things where there's a very low sort of what's called variant allele frequency, or VAF, where we expect to see very infrequent errors. So like, for example, if I want to take a blood sample from you, DNA circulating around in your system, I need to know exactly what that variant allele frequency is. I need to be able to detect very rare mutations. And with this, when we're using the polishing, we're actually able to get pretty accurately, we get down to 5.24% where we expect it to be 5. When we're not polishing it, we were measuring 6. Now we're getting it to 5. We're still working on validating this and enriching this and making this a more portable, targetable assay. So you could potentially use this in a doctor's office, or to be able to check to see what frequency of mutation you actually have, and to inform things and have this be portable and more democratized. Because right now, sequencing is really confined to core facilities. It's not really translatable. We want this to be translatable. But, and as Ken was telling you, there's other advantages to using nanopore sequencing. It's portable. I told you it's a portable instrument. It's $1,000, so you can and be able to move it around. But we're also measuring the DNA molecule directly by feeling it out with the electrical current. What this means is that we're not sequencing it by looking for base pairing of A to T and C to G. No, instead we're feeling the molecule with how the current sort of gently flows around it, how the chlorine ions are flowing around it. It's like an electrophysiological measurement. And so what that means is that we can detect the difference between thymine, cytosine, and 5-methylcytosine, pictured here for those who it may have been a minute since your organic chemistry class. Thymine has an extra methyl group and a carboxyl at the top. Cytosine has an amine and no methyl group, and 5-methylcytosine has the methyl group and an amine. When we run this through the nanopores pictured on the right, we found that you can see different electrical currents for example, GCT-TGA versus GCT-CGA versus GCT-methyl-CGA. By looking at the electrical current and training a model, whether it's an HMM model, as we originally did with nanopolish back in 2017, or a neural network model, as people are now doing with tools like Dorado, we can actually call DNA methylation. DNA methylation is an important regulatory layer that sits on top of DNA and helps to control whether genes are expressed or not. We all have the same DNA all throughout our bodies, but cells in our brain don't look like cells in our feet. They're expressing different sets of genes. It turns out that this worked quite well. Like, we showed that it agrees with sort of more conventional methods like whole genome bisulfite. For alignment, we get a correlation of about 0.937 as plotted on the left in the scatter plot. And you can see that generally we're seeing really good agreement with this sort of bisulfite measurement. But more than that, because we have long reads, we can map to places in the genome that you couldn't map to before. A lot of the genome is repetitive, and short sequencing reads, which we're usually getting with bisulfite sequencing, have a really hard time of mapping and uniquely aligning to the right spot. Long reads are long enough. It's like if you think about a puzzle, if you have a little puzzle and you have lots of little blue sky pieces, you don't know where it should match up. But if you have a big piece of a puzzle, you always have a little piece of an ear or a fire engine or a bird in it. And so you can usually figure out where that should fit in the puzzle. So long sequencing reads map better to the genome because you're more likely to have a unique piece in it that tells you where you should be, to be able to have a fiducial mark to tell you where you should be. More than this, though, that now, in the past like 2 or 3 years, standards are emerging that allow us to incorporate these because the modification data is just embedded in the DNA, and it comes along for free when you do it. There are now standards in what's called BAM files, which are sort of the alignment files, which encode the modification information directly in it. And this is efforts through the sort of file specs and GA4GH and some of these consortia, which encode it into both probability that's modified in the ML call and what the modification is in the MM call. These tags mean that now people can develop downstream bioinformatics tools that are going to operate based on these things, which means that we don't have to make these esoteric file formats. This means that we can actually use things. And an important feature I want to emphasize here, especially for the students, is if you develop tools, if you're thinking about things that other people might want to ever use, you got to think about downstream usability. You got to think about standardization and downstream usability so that this stuff is useful for the entire scientific community, not just for your lab. And so having things like this BAM file spec enables that. And using this BAM file spec, we can analyze both nanopore data and PacBio long-read data because they can both fit into this same framework, and then the same downstream tool can be applied. Here's an example of one of the times we did this. We looked at this cell line HE002. This is this guy who has donated a lot of DNA samples to the scientific community. So he's been sequenced extremely deeply. And in this case, we sequenced them with both PacBio sequencing, as shown at the top 2 lines, and Oxford Nanopore sequencing, as shown at the bottom. And we called DNA methylation, and methylated CGs are shown in red, unmethylated CGs are shown in blue. But because the reads are long enough, you guys all know you have 2 copies of DNA in all of your cells, one from your dad, one from your mom. These copies differ slightly because your dad had different DNA with different single nucleotide variants, with different variants, different haplotype tags basically on them than your mom. With the long reads, we're more likely to run into a place where your mom's copy and your dad's copy differ, which means that you can now separate out the sequencing reads to see this is one allele and this is the other. We did that here at this location, and we see that there's a lot of allele-specific methylation. There's a lot of locations here. This is a location on MEG3 on chromosome 14, which is a long non-coding RNA, where we see that the one copy, haplotype 1, is methylated, and the other copy, haplotype 2, is unmethylated. And we're seeing that same kind of data in both the PacBio and nanopore. So we can analyze the data in the same way, and we have this framework. And using long reads lets us map to repetitive areas, but also lets us split out places where the 2 different molecules of DNA you had were different. So what else can we do with this? Well, we've been working with a group down at Baylor University in Texas on this project, this All of Us project. For those who aren't familiar with All of Us, it's this effort that was originated at the NIH several years ago now to sequence all of us Americans is the idea. And it's supposed to sequence millions of individuals, correlate them with phenotype, and get a better picture of genomics for a more diverse crosscut across the entire American population. So there was an effort a couple years back to try and apply new technologies to the All of Us consortium. Most of it's doing short read, but we wanted to do some long read on it. And so we worked and we selected about 1,500 self-identified Hispanic individuals that we then sequenced with Oxford Nanopore sequencing to about 30x coverage. And we were getting coverage across pretty much the whole human genome, where most of our reads were about half of our data is coming from reads that are about 20 kilobases long or longer. And we wanted to map this and see what do we see from this. And there's a lot of findings we had about structural variants and stuff. I'm going to focus here on what we found from the epigenetic data. So the very first thing we found was that when we tried to correlate from all of these samples, from these 1,500 samples, your so-called epigenetic age versus your real age, we can see a good correlation. What's your epigenetic age? So there's if you look in the popular press, but even in the scientific literature, there's these ideas of this epigenetic clock, which tells you based on your epigenome how old you are, and then you can match it up against how many years you've actually been alive on Earth, and you can see that the correlation here ain't so bad. It's in red. We do see a little bit of a residual when you subtract off what the actual chronological age is versus the epigenetic age, where in our cohort, those who were younger seem to have an older epigenetic age, and those who are older seem to have a younger epigenetic age. So it wasn't quite perfect. But part of that is because the epigenetic age is partially with cell type, because the All of Us study is based on human blood. And so if we're taking a human blood sample from you, we're not solid inside like a potato. We're not uniform. We have all sorts of different cell types inside us. And if we look at blood, you have T cells, you have B cells, you have a lot of neutrophils, you have all these different cell types in here. And so a really talented postdoc, Yi Fu, down at Baylor, used the methylation data that we had to split out and say what different cell types are in there, because there are certain markers that we know identify T cells from B cells, from neutrophils. And you can use the methylation data to split out and quantify how many of these different cell types you have. And we found that according to age, you can see that there's a slight dependence on fraction of the different cells here for like granulocytes versus T cells versus B cells versus monocytes and macrophages. There's a little bit of a different dependence, and the different subpopulations even have a slightly different amount of the different populations. So part of what we're seeing as so-called epigenetic age is actually due to the heterogeneity of the sample. And this is something you always have to think about when you're looking at the epigenome. All of your cells might have, with the exception of germ cells and other mutations, might have the same DNA sequence. But your epigenome, your transcriptome is very different. We also wanted to see, well, are there any other principal components? Are there any other things that we see in the Hispanic population, which we expect to be a very genomically admixed population, that we can pop out that have differences in the methylation? We do see some components that are pretty significant, where we can see that, in fact, some of the principal components seem to separate the ancestry within the Hispanic population, where we see, AMR versus EUR versus African versus admixed. We can see some of the different populations, and some of the genomic variants are helping to dictate how your methylation changes in these blood samples. So there's something about how your genome sequence instructs your cells on how to regulate its genes based on their methylation state. And we can start to see that here. We can even take it further and look for so-called methylation quantitative trait loci, or methylation QTLs. And when we look at that, we found 500,000 uniquely identified methylation QTLs in this data set. Because again, this is one of the first very large epigenetic data sets. Most of the rest of the time, we've had to use microarrays to generate samples of this size. But now, whenever we do long-read genomic sequencing with either PacBio or ONT, we're going to get methylation data for free. And so we're going to learn a lot more about the epigenome from these samples, just by virtue of the fact that the data is going to be there to be picked up and used. And we see that we get a lot of these methylation QTLs from this. Other studies of about this scale, for example, DCODE, which is a study done in Iceland, which is frankly a little bit of a genomically homogeneous population, finds a lot of the same MQTLs that we do, but we found a lot of unique ones in ours, unsurprisingly, because Hispanic populations are generally understudied. So this is kind of fun and exciting. But perhaps the most exciting finding to me personally was this idea of looking at paralogous genes. What's a paralogous gene? Paralogous genes are copies of almost the same gene, but not quite. So during evolution, let me think about it this way. If you're writing a paper and you're about to make some big edits to the paper, you might want to save a backup copy of the paper somewhere. So you have like version 1, and now you save version 2 or new version or something like that. You make some other name for the paper. That's what's happening with paralogous genes. These are genes that are really important for the organism, but the organism wants to try something new. There's a lot of genes, for example, neural development, that are paralogous genes, because we want to be able to try something new, but if it breaks, we don't want the organism to die. And so we make copies of genes that are almost the same gene. And then we decide, okay, actually, this new version is working better. I'm going to shut off the old copy epigenetically, but I'm going to leave it around, hanging out in the genome, because I don't like to throw anything away. I'm a data hoarder. The genome is a data hoarder sometimes. And I want to see which, and so you have these different copies. The problem is these copies are almost the same. So how do we tell which one is which short reads? You can't. You need a long read that's going to align uniquely to these paralogous genes to figure out what's different. And one of the things we see is we see co-regulation between these different paralogous genes. So this is a case where we see SLC22A5, auxin 2, and SLC22A4. They're both solute transporters involved in lipid metabolism in this case. And they're very similar, but they have slightly different functions. And when we see that if you have a SNP in one, it changes methylation in the other one. Not in itself, in the other one. And when you have a SNP in the other one, it changes methylation in this one. So, and they're some distance apart genomically. So the cell or some other regulatory network is saying, okay, I have a variant here. This copy is working well, so I'm going to shut off or do something else to this other copy, and vice versa. So how paralogous genes are actually regulated is a question we're only now going to be able to explore with long reads. And this is a first meager attempt for us to try and show what you can do with this from this kind of a data set. But these data sets are going to start to become available increasingly. I mean, the UK Biobank, a lot of places are starting to generate large long read data sets, which are going to reveal this kind of information. So I've been talking about methylation for a minute, but there's another type of methylation. There's hydroxymethylation. So hydroxymethylation is this proposed intermediate of methylation where you have, normally you have 5-methylcytosine, but that can get oxidized. You can add a little OH on there and you get hydroxymethylcytosine. This is thought to be an intermediate where you're starting to lose methylation away. And we don't see this a lot in cells, but we do see it in tissues. We see it especially in brain tissues where you have a lot of non-dividing cells and you might oxidize in neurons. And then the cells don't really divide, so then it just sits there oxidized waiting for cell division. We wanted to try and see, can we see this? And so we took mouse tissues and we wanted to start to explore this in both brain, in liver, but then we also decided to expand that even further and try and do brain, liver, 2 different brain tissues, liver, heart, and blood. We did long-read nanopore sequencing, but we didn't trust nanopore sequencing. We wanted to also do short-read sequencing, so we used a method from NEB. It's an enzymatic method called EM-seq, and they also have a modification that's called HMC and EM-seq. And we wanted to try and see, how is this working? We worked with an F1 mouse line from Jackson Lab, so we'd be able to do allele-specific behavior. And we started to look to see, what do we see from HMC? So the first thing we saw from an early pilot study was that this is promising. We see that globally, Nanopore reports that there's 70% methylated DNA, and EM-seq reports about the same thing. Globally, Nanopore reports that for cerebellum, we have about 10% of the CGs are HMC. That's a lot. That means that we're going to have some signal here, at least from cerebellum, to see. And happily, the short reads agrees with that sort of global figure. Down below, you can see a couple of examples for that I'm going to zoom in on in a minute. But what I want to draw your attention to is that broadly, the top track, which is short read, and the bottom track here, which is long read, look the same. And this track here, which is long read HMC, and this track here, which is long read HMC, short read HMC, look the same. So we're seeing broadly mirrored behavior between long and short on all these tracks. So short seems to be agreeing broadly with long read. To be continued. We are still in the midst of analyzing data from this study, so I don't want you to be like, your take-home message should not be, this definitely works, I'm done. Your take-home message should be, it looks promising. And you might say, but why do I care? Well, typical methods for profiling methylation don't actually look at 5-methylcytosine. They look to see, is the cytosine modified or not? So, for example, if I measured with bisulfite sequencing ADAM22, ADAM22 is a gene that's sort of important for neural development and for neuronal function. You can see here, this is the methylation profile for 4 of my tissues, for cerebellum, cortex, heart, and liver, and it looks pretty uniform. All the tissues seem to have basically the same methylation profile. But no. If I now break it out and try and look at methylation, 5-methyl-C as colored by the solid lines, and HMC with the dotted lines, you can clearly see that the brain tissue actually has lower 5-methyl-C and much higher HMC in this region. So we can see that there are differences that were not visible to the previous technique that we're now able to see by splitting out HMC versus MC. What does this mean? I don't know yet. We're still trying to look to see and understand where all these differences are. But my point to you is that now we can start to observe them. And they clearly are significant and consistent because this is a representation of the median behavior of all 5 tissues at once, of all 5 mice at once. And so we're seeing that this is replicated across different biological replicates. It's not like a one-off. It's not like a variable thing. This region definitely has an enrichment for HMC and a depletion of 5MC. And similarly, it's not just brain. Even though liver has a much lower level of HMC, ApoB, which is an important gene for metabolism, has a significantly higher level of HMC in liver tissue, where it seems to be pretty low. So there's something to looking at HMC. We're generating this relatively large data set that we're going to share with the community as soon as we're done making it and doing a first pass on it. And I hope that it's going to be useful to start to think about what is HMC actually telling us about how methylation is regulated and therefore how genes are regulated. So methylation is one piece of the puzzle, but then there's also this piece where we care about how easy is it for proteins or transcriptional factories or other things to get to the DNA, this idea of chromatin accessibility. How DNA is organized in the nucleus depends on a lot of factors, but like it can either be tight in and closely regulated, or it can be like more broad. What we wanted to do was see if we look at DNA methylation here, if we look at chromatin accessibility, can we see with long reads, can we see and answer some of these interesting questions? So we used a method that we called nanonome, where it's based on a method from Kelly et al. Published almost 13 years ago, I think now, where what she did was she decorated accessible chromatin with a different methyltransferase. So she's encoding using modifications into the DNA molecule itself, something about the state of the cell, something about how easy it was to go and touch the DNA using this methyltransferase. So she's painting like with a stencil on the DNA with a methyltransferase and seeing what she can see. As I said, my student called it nanonome, and so he has this little picture they used as an icon to represent that. But what happens is this GPC methyltransferase decorates here, and then we still have the native DNA methylation at the same time. So that means that now I have multiomics on each individual read. On the left, you can see I have DNA methylation, which is the same thing we've been talking about up to this point, and this is at a location called PIM2, which is has two different clusters of reads. They have an inactive allele and they have an active allele. And you can see that PIM2 is unmethylated for a portion, and then methylated over here, and then unmethylated again. And you can also see over here on the right, this is the chromatin accessibility signal. We see a distinct area of accessible chromatin, so you can get the polymerase on and start transcribing the RNA on this active allele, and the inactive allele doesn't have any space like that. There's no place for it to come in and start getting to work. It's all closed off, so that allele can't operate. We use this, as Ken was saying, we use this even on the new T to T genome. This is part of what we found from the T to T work, where we were able to look even inside the centromere. So the centromere, if you ever think about the picture chromosome, that X-shaped chromosome, the center of that X-shaped chromosome is the centromere, and that's where the kinetochore forms, and that's when you're doing mitosis, how the cell separates out the DNA chromosomes from each other. But that area is basically all repeat. It's very difficult to probe without long reads. So we were able, as part of the TAT project, we were able to probe that for the first time. And what we saw was we could see this so-called centromere dip region that we coined, where it's an area of low levels of 5-methylcytosine. And it's also an extremely inaccessible region based on the chromatin accessibility, as shown here by this Z-scored blue curve. And we found that this is probably where the kinetochore is actually binding, inside the centromere, this sort of undiscovered country, this dangerous, treacherous area that's just full of repeats. We can explore it with long reads, and we could see that we think that this is the place where the kinetochore actually touches down, binds on, and helps split out cells. So if you think about lots of Mendelian disorders, like where you have trisomy or other places where you have chromosomal defects, understanding what's going on in the centromere might be critically important. And as like Adam Philippi and Karen Miga, who led the TAT project, as Karen keeps working on generating more TAT scale genomes and Adam keeps assembling more TAT scale genomes, we're going to have an opportunity to really understand more and get more functional insight using this. Thinking about the segmentally duplicated paralogous genes I was talking about, thinking about the repetitive areas that we can now map to, we're going to be able to explore these new things. And we can actually use how big those chunks of inaccessible versus accessible chromatin are to say something about what's even bound there. So typically, DNA is wound around these protein assemblies called nucleosomes, which are assemblies of multiple histones at once. The DNA winds around them. Those block off something that's about 130 bases of DNA sequences. So we see very distinct signal at about 131 of inaccessible runs of DNA on a read. We also see sometimes we get a dinucleosome where you had two of those things that were so closely stuck together I couldn't label anything in between, and those show up at about 300 or 260 or something like that. But sometimes we see, especially if we look flanking this other important protein, CTCF, flanking this binding site, we see a smaller footprint. We see something that's only about 30 or 40 bases long of inaccessible DNA. And so if we look over here, at this plot where we're looking at the accessibility runs and the chunks of accessible, blue, versus inaccessible, sort of peach, DNA, you can see that in a big accessible region, I have this little area that's inaccessible. That's an actual protein sitting down. We can actually do protein footprinting, and we can count how many molecules are bound by a protein here by just counting how many reads there are versus how many aren't bound. This allows us to actually get at least a quantitative measure of, in a population of cells, in a population of molecules, what fraction at a given time when we did the measurement were actually bound. So for example, over here, we can see a region where we had high binding, where like 85% of reads actually had that footprint in place. Whereas on the right, we can see that only say 2% of the reads had that in place. Now, typical methods like chromatin immunoprecipitation or ChIP-seq, or even cut and run, lose some of this quantitative nature because they're doing a lot of PCR and they're amplifying, so you can see peak or no peak, but the strength of the signal isn't necessarily as indicative. As shown here, we have a lot of places that have peaks for ChIP-seq, but the variability in the fraction of reads that are bound is pretty strong. So we think that being able to use this to count the fraction of reads that are actually bound could be a powerful metric. And I'll add that my friend Andrew Stagakis at University of Washington has a method called FiberSeq, which does a very similar thing. And in some ways, it's easier than even what we did with NanoGnome a couple of years back. And he's trying to commercialize it with PacBio. These methods can be really powerful to look at accessibility in hard-to-map regions, to count and be quantitative about what's accessible or not. But sometimes you might want to use a more targeted method. Sometimes you want to spend your sequencing reads a little bit more directly. And so we might want to use a method like something developed originally by Steve Heinekoff at the Hutch called cut and run. Cut and run is this method where you take cells, you have an antibody, and you have something fused to them that's called an emanase. The emanase will then, the antibody comes in, recognizes a protein, and then the emanase will cut the DNA flanking the protein, and now that DNA floats free. And you can sequence the DNA that floated free and figure out, well, where was it? You know, we can align it then to the genome and see, oh, so this protein must have been where my floating free DNA was. So my postdoc, Paul Hook, looked at this and he said, well, maybe I can just not do any amplification at all. Maybe I can just throw this directly on a nanopore sequencer, and then I could keep methylation and I could do all sorts of stuff. And I said, well, that's a really stupid idea. That's never going to work. And he's like, well, I'm going to try it anyway. So he did, and it worked. And I got to tell you, trainees especially, that's the best feeling as a PI, is to have, that's the whole reason that we're here at a university, is to have you guys try something that we think is stupid from your, but you try it from your perspective, and it works, and you found something new. That's a great feeling. I'm serious too. It works really well, actually. We can do it without amplification, and then you have that, again, that portable cheap sequencer that you can do to look at protein DNA interactions right at your bench. You don't have to send it out to a core facility. You can do this just locally. It works just as well as doing it at Illumina at a large core facility. But we can also do it without amplification. And so without amplification, I told you before, we get to keep the DNA methylation data. So what you can see here is, here's a peak where we're looking at H3K4 trimethylation. This is a mark of active DNA promoters. And you can see, here's the DNA sequencing reads I get from Illumina. Here's if I amplify Oxford Nanopore sequencing data, and so we see this nice little peak. But here's when I don't amplify Oxford Nanopore data, and I also, here's the reads here, and you can see, not only do I know that that place was marked by H3K4 trimethylation, but I also know its methylation state. I can tell that it's an active, I know that it had H3K4 trimethylation, and also that it was an unmethylated promoter. Turns out also, because I didn't do any amplification, I wasn't subject to the shortening that typically happens from doing PCR, and I get a little bit longer reads, which means that not only do I get a little bit better mapping, it's not as good as the super long reads I was showing you from FiberSeq and NanoGnome before, but it's good. But I also can even phase out the reads, so I can do allele-specific protein DNA interactions a good fraction at a time. And like with Illumina sequencing, I can do it only about 5% at a time. But with unamplified cut and run, I can do it about 25% of the time. I can split out the reads and assign them to one allele versus another. And this is even on a really boring, I think this was on GM12878, which is a really boring white woman from Utah. I'm not judging her as a person. I'm saying she doesn't have a lot of variance. And so we want to, so if we looked at a much more interesting sample where it's more heterozygous, we might be able to see even more variation. So what if I wanted to do even deeper multiomics? What if I want to do chromatin accessibility, DNA methylation, but I also want to label on there what protein is bound where. I wasn't content with just the footprinting. I want to do something else. We've been working with a company down in North Carolina, Epicipher, where they're adapting a method that's similar to DMeloseq from Aaron Streets at Berkeley, where we're trying to commercialize this idea. Because, again, I want it to be usable for everybody. I don't want people to have to make the proteins from bacteria. I want them just to be able to have a kit and use it. And so what we're doing with them is this method that they've called Katana LRS, where you have an antibody fused to a methyltransferase. I told you about antibody fused to emanase, where it's cutting. Instead, now it's an antibody fused to a paintbrush. So now it's painting on a modification. You can paint on the chromatin accessibility modification, and then you can paint on a different modification for protein DNA, and you might be able to measure that. We're doing it one step at a time. So what we're doing so far is just trying to measure, can I mark on here where, for example, H3K4 trimethylation is using methyl-6-adenine. And so I'm showing you this here in green. This is a mark that isn't native to human cells. And so we don't think that there is any M6A normally in humans. And we've tested that also with the sequencing. And when we try and label, we see that the cut and run shows us a peak here. And our methyl-6-adenine also is showing us a peak. It's a little noisy. Nanopore isn't as good at calling M6A, and we're still working on validating some of our experimental conditions, but it seems to work. And again, we are getting 5-methylcytosine data from the same time because this comes along for the ride. Want to layer on chromatin accessibility. When I look at metaplots, it does seem to be for, if we look at ranking of the strongest bound H3, strong M6A for most of those. So it's working okay. It still needs a lot more validation and optimization, but I wanted to share it with you. So I talked a little bit before about this idea of splitting out the cells from samples, and I want to expand on that a little bit and prove to you that how well it's working, also as a transition to talking just generally about the heterogeneity of tissue samples. So for cell type deconvolution, this is the kind of data we might get. So these are individual reads where we're looking at DNA methylation shown in red and unmethylated CGs shown in blue. What's combined here is two different cell types, HET116, which is a colorectal cancer cell line, and HE002, which is a lymphoblast line. And what you can see is that we see 4 different clusters of reads when we just try and de novo cluster them, not knowing anything about the cell type here. We see an allele here on HET116, so I have the actual assignment over here too, which is telling you that clusters 0 and 1 come from HET116. This entire area is unmethylated. Only this portion of the area is unmethylated. And then in 002, that same portion is unmethylated, but then you can see there's also differences over here, which you might not be able to see with a short read. With a long read, you get the whole enchilada. And then you see cluster 3 is different again. So we can actually separate these out pretty effectively with just a de novo clustering approach. So what we wanted to try and do was, let's actually take some blood and let's be really careful about this. Yilei was doing stuff where he was using existing cell type atlases. We wanted to say, compare the same blood sample that's well mixed. Let's do single cell RNA sequencing to get a sense for what different cell types are in there. And then on the same sample, let's do DNA sequencing with Nanopore and see how well our quantification metrics match up. We targeted our Nanopore sequencing using something adaptive called adaptive sampling, which I do not have time to get into, but I'm happy to talk about later, to focus it on regions where we expected to have cell type specific differences. And we wanted to see, how does our quant match up? It matches up pretty well. What you can see here is that in blue, I'm showing you cell type specific quants from 3 different samples based purely on methylation. And in orange, I'm showing you the cell type quants based on single cell RNA sequencing. Single cell RNA sequencing is looking at way more cells. We only had something like 100x coverage, so we were only at best sampling 100 different cells from single cell, from nanopore methylation sequencing. And the single cell RNA sequencing had like 1,000 or 10,000 cells that we were looking at. So we had way deeper sampling there. But you can see that these bar plots are generally showing pretty good agreement. So we think that we're going to be able to use the long read sequencing to actually do what I'm calling cell type deconvolution. It's not a new idea, but to be able to figure out from any given sample you sequence what your proportions at least of cells are, and even be able to phase on there a genomic variation associated with a cell type, because we can tell which read came from which cell type as long as we hit a cell type specific DMR. So how often are we going to hit a cell type specific DMR? Pretty often. Cell type specific DMRs occur pretty frequently in the genome. This is a sort of cumulative distribution plot where I'm showing you the frequency at which these different variants occur. And what you can see is that this is on a log scale. So this dotted line here is how often we're going to hit it with short reads. You can see only about 30% of the time does a enhancer come within range, or maybe 40% of the time does an enhancer come within range of a short read of a cell type specific DMR. But if I have like a 10 kb long read or even 100 kb long read, I'm almost always going to hit a cell type specific DMR. So if I have nice long reads, I'm probably going to be able to tell you the difference between which cell type it is. Maybe not if the cells are like killer T cells versus memory T cells, or cells that are very similar to each other, so they have very few epigenetic changes. But if I'm looking for example, in a tumor sample, if I'm looking for differences between a tumor cell and a cancer-associated fibroblast or a tumor-associated macrophage, I'll probably be able to tell those apart, and I'll be able to know these were the cancer cells, these are the reads that originate from cancer cells, and what's going on with them. So that all said, we do to try and look at tissue differences? Well, the most common way nowadays to try and look at tissue heterogeneity, I'm going to argue, is to use single-cell RNA sequencing. Single-cell RNA sequencing, as probably best commercialized by 10x Genomics, even though works like this, where you actually, as you isolate cells physically into droplets with beads, and you put code on, as shown here in green, and a unique molecular index. So after you do all the PCR you're going to do, you don't bias your quantification. But then you only get a read from the very end of the molecule. And I say that's not enough. I say I want to sequence the whole transcript. I want to see it from tip to tail. I especially want to see it from tip to tail if I'm thinking about things like cancer. So one of the first studies we did with this, with Theron Palmer, who's now at the VAI here in the great state of Michigan, up in Grand Rapids, is looking at pancreatic cancer. Pancreatic cancer is a big thing at Hopkins. We love, nobody loves pancreatic cancer. We work a lot on pancreatic cancer at Hopkins. And so what we're doing there is we take a surgical resection, we took organoids to make it so we had enough sample to really be exploring it. And we did single-cell RNA sequencing. We did this on 5 different organoids. And what I'm showing you here is a picture from KRAS. And this is long-read transcript sequencing of KRAS. And you might say, big so what? Looks like I'm seeing one transcript isoform, there's no cool splicing. Here's so what. If I was doing short read sequencing, I would only see a little bit of this part. But for KRAS, there's an important mutation which is diagnostic and informs your therapeutic treatment. And if I have a complex cancer like pancreatic cancer, I might want to see what the mutation distribution is in tumor. I want to see in this cell what's the mutations, in this cell what's the mutations, even in the coding sequence. And if I can sequence the whole transcript, I'll know what mutations were in that whole transcript. So when I can zoom in on that red box, I can see for the different organoids, different distributions of different mutations. I can see either a KRAS G12D mutation in red or G12V mutation in green, and I can see the variant allele frequency within the organoid, because the organoid it's patient-derived, and so there's a bunch of organoids spread out from the different cells, and so we can see what the variant allele frequency is, even from the different organoids that we had in our sample. The end goal I have here is to take actual tumor samples and be able to regenerate a cancer phylogenetic tree from this, because I could see, here's all the mutations. These cells have this cocktail of mutations. These cells are probably precursors because they only have a subset of mutations, and I can try and reconstruct the timeline of the tumor from doing this. I think that this could be powerful to do that. I know it's just the coding genome, which maybe is less interesting, but I can still look at it by looking at the transcripts. We can also do simple things like figuring out HLA type from cancer, because we get the full long read from this. And we found that we got pretty much the same HLA types from our long read approach that we showed on the right there to the short read approach using something called optotype. We did have one mistake, and that was we got one base wrong. We got it wrong. We're still working on figuring out how to fix that, but that's where we are with that. So that's fine, but Winston, you weren't really talking about different cell types there because it was organized. That's true. So let me tell you about a slightly different story where we tried to understand how learning works. I mean, we're at a university, we always care about how learning works, but what we wanted to do here was we wanted to take mice, expose them to a context, that black and white checkered thing, and then expose them to the context to give them a foot shock. Then we immediately sack the mice, harvested their brains, looked at their hippocampus, where memory and learning are sort of centered, and we wanted to see how does that affect RNA? How is that affecting splicing? How is that affecting what genes and what types of genes you're expressing? So it turns out we can separate out different cell types pretty easily from single cell long read sequencing. Shown on the top is when we do this with Illumina sequencing on the same sample, and on the bottom is Oxford Nanopore. We get the same cell barcodes. We get generally the same cells are identified. There are a couple that we saw with that we didn't get with ONT that we got with Illumina, and that was just a sequencing depth issue. Most of the time, there's very good correlation of what was assigned. Interestingly, we see that there's a shift in the polarity of response, depending on if we got naive mice that weren't exposed to anything versus mice that were exposed to the context versus mice that were exposed to the context and shocked. We see different sets of genes that are actually being expressed, and there's a shift in the polarity depending on the type of cell. So you need to do single cell sequencing on brain to figure out what cells are responding to what. Pyramidal neurons, as shown on the left, are more seeing, I saw a black and white pattern. Oligodendrocytes, by contrast, seem to be seeing a difference in behavior depending on if they were exposed to a context but not shocked versus shocked behavior. But here's the cool part. This is the reason why I wanted to do long read. We see lots of occasions where we see, and so in the dark blue, especially for specific cell types, where we see no difference at the gene level of expression, but we do see differences at the isoform level. So different isoforms are actually being expressed that would have been invisible if we'd done only short read sequencing. So we want to understand what's happening at some of these. Here's an example where you wouldn't have been able to see this before. This is a S100A16. It's another sort of ion transport protein. Here's the Illumina sequencing reads from the sample where you have them all piled up here in orange over in the 3' area. And here's the long reads. When we see long reads on specific cells, you can see astrocytes have this isoform where they have this exon. And then oligodendrocytes and inhibitory neurons have this other isoform. So we can see isoforms that are specific to cell type very clearly by doing long read single cell sequencing. Here's another fact. If we looked at CA1 pyramidal neurons, which is a really important part where we think we have engrams that are actually encoding the memory. So there's a subset of cells here encoding the memory. Let's look at a specific isoform, BAIAP2. And we look at BAIAP2 in CA1 that are either context exposed or even unexposed, that get that black and white pattern. Let's not even worry about the foot shock for now. There's a different set of isoforms for the C plus and U plus than the naive. Let me zoom in on this a little bit. So if you look at C plus here in orange, U plus here in green, and naive here down in blue, you can see very clearly that you're getting two different isoforms of BAIAP2. And you might say, so what, Winston? Why do I care that I have this different isoform? Well, it turns out that what you've done is you've exposed, by having this different splice, you've exposed a different binding motif which acts to change where the protein ends up in the neuron. So this difference in splicing moves the resulting protein to synaptic complexes. So it's part of what's controlling how the synapses are forming. I am not a neuroscientist. I just would play one on TV if they would let me. But this is what my collaborators and colleagues have reassured me, that this is the kind of hot sauce that they're interested in, is trying to see and understand what splicing might be doing functionally to change how the neurons are activated in behavior. And so now with generating this kind of data, we can start to explore this. But in the brain, you gotta parse out the different cells. But more than that, you can't just parse out the cells, because sometimes the cells behave the same. Sometimes the cells might be the same cell type, but might behave differently depending on where they are. And so we want to do stuff where we're actually also encoding the location of where in the tissue it was. And we can use methods like spatial transcriptomics to do this. This is a method sort of commercialized by 10x called Visium, where they encode using little barcodes on a slide. You slap down your tissue piece on a slide, and it's spots that have a size of about 55 microns with a pitch of about 100 microns apart. So it's not very high resolution, but it's good enough for government work. And so you can look at this, you get the RNA, you encode the barcode into the RNA depending on where you are. And I'm here to tell you can do long read with this too. So the first thing I do is I trust but verify. I don't ever trust that anything is working from any company ever for any reason until I've tried it out myself. And so what we did was we made what I call a spatial barnyard. So we took a piece of human tissue, we slapped it on the left, took a piece of mouse tissue, we slapped it on the right. Humans and mice are a little different. And so their transcripts are different. And so by alignment, we can determine if this read should have belonged to a human or should belong to a mouse. And that is color-coded here, where bluish green here is supposed to be mouse reads, and orangish red is supposed to be human reads. And we see that, in fact, when we try and demultiplex, reads that should have been underneath a piece of human tissue showed up as human reads, and reads that should have been underneath a mouse tissue showed up as mouse reads. So our demultiplexing sort of works. The spatial part seems to work. We're getting long read data that works. Working with colleagues at the Lieber Institute, including Kristen Maynard, and Kerry Martinowicz, we took dorsolateral prefrontal cortex from postmortem human brains, took some slices, and we threw it on nanopolar sequencing from 4 different samples, and we wanted to see, do we see differences in the layers? They have already shown, by the way, that you see differences in transcription between the layers with short read sequencing, but we wanted to first confirm that we can see that. So we have the short read data up above, we have long read data down below, and you see very clear differences. When you just try and cluster this de novo, you see very clear differences in the transcriptional activity between layers. This is partially due to white versus gray matter. It's partially due to a lot of other differences between these layers, but we can very clearly get the same kind of clustering that you're observing here between short and long. We do where there's differences. I don't know if this is as clear. It looks like it's pretty good, where you can see that the sort of dark or more filled in dots are places where they don't agree, but you'll notice they're always on the border. And I consider that just sort of technical variation and noise. Like it's the ref at the football game. Are they calling it in or out? Is it a touchdown or not? I don't know. Let's go to the videotape. You can see that this is a tough call to make. So I consider this to be pretty good cluster assignment. So now let's shift to talking about gene expression. With spatial transcriptomics, let's just look at, we're still in easy mode here because this is relatively new data. We're looking at white matter versus gray matter. Plotted on the top is the dark blue is showing you where I think there's white matter. Everywhere else on the slide is gray matter. And you can see that I see GFAP, which is a marker for white matter, has very clear differences in gene expression shown on the bottom between places that are white matter versus gray matter. It's very bright here, it's not so bright here. That's because GFAP is primarily expressed up here in the white matter. And that's fine. But now, what about isoforms? Can I tell what isoform it is? It turns out I can. I can figure out that the GFAP isoform that's actually expressed in that layer, in the white matter layer, is GFAP 214. I don't have to worry about being 203, 225, 201. I know exactly which isoform it is. I can tell by using the long read sequencing. We're still trying to see if we can tease apart isoforms that are specific to different layers. We have some new data, but that I'll tell you is starting to look like we can see some signal there. But I will also confess to you that part of the processing of the Visium data means that a lot of the RNA is broken. It's also possibly partially due to the fact that postmortem human brains are often not super great RNA. For some reason, some time has passed since the person died and the brains are harvested. If I'm going to do anything, honestly, I'd probably try and prefer to work with mice because I can harvest their brains much more rapidly after death, and so I'm going to get better RNA. But this is also more interesting because mouse brains aren't very representative compared to human brains, which are pretty complex and interesting. I would convince you that long read sequencing has some cool applications. I will tell you, especially trainees in the audience, don't be swayed by the new shiny object. Use the technology that's going to make the most sense for the experiment you're doing. Do the simplest possible experiment that you can do that will answer your biological question. But sometimes that might be long read sequencing. With some of the new tools and technology, we can look at things we could never look at before. And I think it's kind of exciting. I want to especially thank some of the students who have worked on this, which are highlighted below, especially Sheridan Cavalier, who did some of the mouse work, Hope Eden, who did spatial transcriptomics, Courtney Hall, Paul Hook, Isaac Lee, and Luke Marina, and Theron Palmer. And of course, my funders. Thanks. I'll take a couple questions.
Thank you so much for the wonderful talk, question.
Thank you for your nice talk. I have 2 questions. I'm just curious whether the methylation of cytosine is an irreversible event or not, and it is related to epigenetic ages or not. And the other question is about the isoform in the spatial transcriptomics. I'm just curious.
Could I answer the first question first? So I
Yeah,
forget
you
it?
can do that.
DNA methylation it's not necessarily reversible, but part of the hydroxylation is a way to do active demethylation. So you methylate the DNA. Now I have a methyl group on there. To demethylate it, what I have to, semi-conservative replication of DNA will basically mean that the new molecule won't be methylated. And so I have to then remethylate the other strand. Or you can hydroxymethylate, and then there is an active pathway that will then strip it off and replace it with a fresh cytosine. But we think that's more rare. We think it's more passive demethylation, where you oxidize it, and then after cell division, it doesn't get recognized and maintained, and then it's diluted through cell division. Your next question was on spatial transcriptomics?
Yeah, isoform in the spatial transcriptomics. I'm just curious, when you quantify the isoforms in the ST data, you rely on exon count or inter-exon junctions?
When I'm quantifying the isoforms, I'm actually aligning the read directly to the possible transcriptome. And I'm using a tool called ORFISH from Rob Patro at UMCP, which does sort of an EM algorithm to try and do assignment and figure out which read this is most likely assigned to. If I have to go back to, let me show, it's probably most clearly illustrated here. So look here, this read here in green, this read is this, and this. It's all one piece that touches all three of these exons. So through alignment alone, I know that it's that isoform because I have the entire intact molecule that has all three of the exons at once. I'm not inferring, I'm not looking at junctions. I have the whole transcript. Does that make sense?
Yeah, thank you.
You're welcome.
Thanks. Hey, congratulations. Really nice work. Very impressive stuff. I had two questions also.
Okay.
Sorry. The first one is about the footprinting work, and it's beautiful. And CTCF looks just fantastic, but it's got like footprinting is based on residency time. CTCF has the highest residency time by like order of magnitude of anything else. Do you see it with other TFs, or is it just the CTCF?
So we use CTCF to look at it. I think that the residency time, so Steve's referring to the fact that, things go on and off. There's equilibrium, right? And so I think one of the things that I actually have cooking in the lab is to try fixation and then methyltransferase. So fixation is going to lock the proteins down and cross-link them, and then I might be able to see the footprint, but it's going to change everything. This is the question, the age-old question of native chip versus cross-linked chip and all these other things. Short answer is I haven't looked at transcription factors. We saw this, it was beautiful. We put this as a figurative paper, we moved on with our lives. But I think you would be able to see it, but it would not be as clean as this because you're going to see different frequencies. But what you would see is you would still see the accessible chunk, and then some of the times you'd see that, but it wouldn't be as instructive as to the fraction that might have been bound if I had taken a different time point. So I think we might need cross-linking, and that's actually something we're working on. We've been talking to Sebastian Pott, also at University of Chicago, about that very thing, because it might also screw up everything to do fixation. So I'm not sure. What was your second question?
Cool. Yeah, thanks. The second question was about the MQTL mapping. So that's a really, really interesting example. And as you're aware, when in the QTL mapping community, anytime you look at paralogs or orthologs, they're the mapping. And so the long reads probably get around this, but there's also the genotyping is susceptible to the same types of biases. So if it's WGS, short read on the genotyping, there could also be mapping bias happening there. And I wonder how
we did the SMB calling actually with the long reads too.
Okay.
So we have both PacBio and, I didn't say, but we have both PacBio and ONT on this.
Great.
And the only reason this paper isn't out is frankly politics, because it's all of us. And if anybody in this room knows about all of us, they know why it's taking so long. Yeah. And but we also called structural variants, we called SMVs, all from this long read data. We checked some of it with spot checks with the short read, because these are also short read samples. But these SNPs that are called here are called from the long read data.
Cool.
So at least I'm not saying you're wrong. Yeah. I'm saying I don't think it's answering any problems.
Yeah, it's just that these are so highly enriched for artifacts that it's quite striking. And I wonder, what do you think's going on biologically here?
I mean, I think I anthropomorphize everything. So I really think about it like it's me writing my grant, and then I decide to cut out big chunks of the grant and rearrange things, but I've made a different copy over here. And if it doesn't work, I go back to the old copy. I really think about it as evolution is, Mother Gaia coming in and saying, oh, let's move this gene over here. I'm going to screw around with this one. Oh my God, that's terrible. Let's turn that off. Let's leave this one on. And allowing us to have some level of redundancy in our genome in case that's how I'm picturing it. The SNP calling and the mapping thing is a whole different ball of wax. And I do think that there's a lot of issues with artifacts. I will also comment as a final note, we are trying straight up assembly of all these individuals, which honestly from the long reads isn't that much harder. So we're doing personalized genome assembly from it to see how that compares our SNP and SMB calls. I'm less involved there because it's not as epigenetic-y, but that work is ongoing at Baylor.
Thanks.
No problem.
Hi, Dr. Tim. You mentioned that ONT can be used to study allele-specific methylation. I'm currently working on a project on allele-specific M6A modification. Since ONT modification detection still has relatively high error rates, so could you share some specific strategies for distinguishing true allele-specific differences from technical noise?
Let me just make sure I'm understanding. You're talking about M6A calling error rates, or are you talking about the SNP calling error rates?
M6A calling error rates.
Yeah, M6A error is still quite error prone. What we're generally doing is we're using coverage to overcome that, and we're being sort of stringent in our filters. So as you know, and as I sort of was explaining to the room earlier, you can set different probability filters for saying, I want to be dang sure that this is modified. Only if I'm 99% sure that it's modified, tell me it's modified. Otherwise, I don't know what it is. Or I can say, I can be much more promiscuous, and I can say, if I'm 80% sure it's modified, tell me it's modified. So I would try a sliding filter there. I would think really carefully about controls for sequences and external validation. If you have regions, I would probably do an approach like this, where I start with some sort of low coverage sequencing. I set my setting pretty promiscuous, and then I say, what regions are suspected allele-specific M6A locations? Then I would go back and I would do some form of targeted enriched sequencing. Maybe it's adaptive sampling, maybe it's something else, to really sequence those locations very deeply and see if they still work when I set a stringent setting, but I have much higher coverage. That's one strategy. Another strategy you could employ, PacBio's M6A calling is way better than ONT's. I would strongly consider instead seeing if you can get PacBio data that would then validate your ONT results. Might be challenging depending on sample and funds availability, but that you could look at existing PacBio data and see if that bears up or validates some of your findings if your sample isn't too specific. So those are sort of the strategies I employ. And I will comment, I'm happy to talk about this too. I'm easy to Google. There are no other Winston Timps. So like if you want to send me an email, I'm happy to follow up. I think you're next.
Thanks for a great talk, Winston. So when you introduced the UMIs, you had a slide on VAFs and you had twist versus ONT. I'm just
I
wondering
didn't really talk about that. So it turns out that how you make the UMIs matters. So ONT, I was saying that the accuracy is good, and it is good, but we want the UMIs to always be uniquely identified properly. Twist makes these short little UMIs that aren't really designed to take into account ONT's picadillos. There are certain differences in sequence that are going to be better than others. The ONT UMIs I'm calling here are actually Mads Albertson's UMIs. Mads Albertson is a guy in Denmark who developed some of these approaches to try and use UMIs to correct for 16S sequencing on samples long ago being 5 years ago. But what he does is he does like YYRR. So he's doing either pyramine or pyrimidine. He's not doing homopolymers. He's not doing random NNNs. He's doing more specific combinations of sequence that he knows are going to get base called pretty effectively. And he's making them nice and long because ONT doesn't care about length. Twist doesn't do that. So we tried to use Twist because here we're trying to do HYBE capture paired to UMIs so we can capture out from serum anything we care about. That didn't work. So instead, now what we're doing is ONT UMIs. That works, but now we have to still get the capture part to work. So this was, I didn't really get into the weeds of it, but thank you for the question.
Yeah, I mean, just a follow-up on that is, could you use now training networks or neural networks to sort of look at this and see what could even be a better
Yes.
UMI, right?
Yeah, so part of, I didn't say, but we also have this whole story where we're trying to encode like pictures or words, like, DNA data storage, right? We've thought a lot about error checking and how to best design these. But to be honest with you, the person who's done this best is Jeff Navala at UW. He has this thing called MALBITS, where he tried to use from the pure electrical data, he tried to figure out what is going to be the best sequences that will distinguish themselves from each other. He even wrote UW out in electrical data, which I thought was cool. And so he has that whole little story. I think he's done it the best of anybody so far that I've seen published. So I would look at his MALBITS paper, which is, I think, from 2022. But absolutely, we know what the predicted electrical signal is. We know what the k-mers are. We could try and do our best design. But since YYRR works okay, I don't know that I need to. So it depends on how dense we want the information stored.
Winston, we have questions from the Zooms. So one question is, what's the typical numbers of the UMI in your spatial transcriptomics data?
The typical numbers of UMI. So yeah, we're sequencing the spatial transcriptomics quite deeply. We're usually getting something on the order of 6 reads per UMI or something. We really went hog wild pretty deeply on the sequencing, where we did like 3 Promethean flow cells per sample, because we wanted to make sure we were sequencing deeply enough to pick up as much as we could. We could certainly do less. Not pictured here is experiments where we're trying to do hybridization capture to enrich for genes that we care about to better spend our sequencing dollar. But the answer is, anywhere from 5 to 10 usually.
Good. Well, myself have a question.
Sure.
Now you can detect not only the methylation, but also the other epigenetics marks like chromatin accessibility, the protein-DNA interaction from the single DNA molecule. Have you ever examined their interplays, the crosstalk between the epigenetics marks?
Yeah, so I really want to do this more carefully, but I think you have to do this with a time lapse. I think you can't do it from a static sample. I think you have to take a sample and watch what's happening over time. I was talking to somebody today about this too. We have to look at, like, as a cell's being reprogrammed, as a cell's going through the developmental pathway, as a cell is transitioning from one transcriptional program to another, what's happening to it? How is it changing? Because in this static quasi-steady state, I don't expect to see something like that. But I expect that, for example, chromatin will change first, right? And then methylation will change later. And so I want to watch that happen. I want to take like MCF7 cells, like some breast cancer cells, which are normally epithelial, and hit them with like estradiol and cause them to undergo epithelial mesenchymal transition, then watch what's happening to the epigenome. I asked NCI for money to do this, and they wouldn't give it to me. So to be continued if I can find money to do it, because I want to sequence it deeply enough that I can actually answer your question. So yes, and the more we can layer stuff on, the better, because I think we're going to be able to see a lot of power. My secret dream is not just to look at this. I want to do stuff like what Heinekoff has done recently and look at where Pol2 is, because I want to encode into DNA where active transcription is. Because all epigenetics tells us is something about what the cell thought it should be regulating. I want to see what's actually being transcribed, but I don't want to measure the RNA. I just want to measure the DNA. And so I'd like to be able to get at Pol2 too. That's it's tricky. There's antibodies against it, but it's tricky. So we haven't gotten it working yet. Thanks for the question.
Oh, we have one more question over there.
Okay.
The very last question.
Sure.
Sorry. Have you had a chance to look, because you have pretty large cohort for some of your studies, how often DNA methylation is heritable and how consistently?
So heritable where, as in the parents have DNA methylation at a loci, and then the children also have methylation at a loci.
Yes.
So this is sort of what an MQTL is getting at, because the parents and the child also will have sort of the same genomic variant, which then drives it. If you're asking for heritable methylation that doesn't seem to have a QTL, we haven't looked at that specifically among families. But I will tell you this: everyone in this room's blood cells looks more similar to each other than their blood cells looks to their brain. The biggest difference we see epigenetically is between different tissues, not between individuals. And so I don't want to say we're reaching, but we're reaching to look for differences between the methylation state in these individuals, because most of these, all the samples, the methylation looks pretty much the same across the samples. There's small changes in aging, as I was referring to. There's a couple small changes depending on genomic variants. But most of the time, your T cells, your monocytes look just like my monocytes. And, we're different. And yet we're not that different. Now, I have a couple SNPs that are different than you, so some of my methylation is different. I got those SNPs from my dad, so I did inherit them, so to speak. So yes, kind of, but we have not, I don't think it's been observed to see epigenetic inheritance across transgenerational epigenetic inheritance is not something we've observed or have looked at in this cohort. I think it's going to be real hard to look at. Like, it's going to be a subtle signal and hard to pick up. Thank you for the question.
No, thank you.
Thank you so much, everyone. Thank you so much for
My pleasure.